And I also want to take this time to thank the organizers for inviting me to come here and share my work. I think it'll become apparent that a lot of our work is sort of going in a parallel direction to that of Dr. Greenberg's. And in a lot of ways, this is going to be a little bit of a step back to really dissect the contributions of the different acyl chains of cardiolipin for mitochondrial function. So this is the biosynthetic and remodeling pathway for cardiolipin in yeast, and yeast is the model that we've been primarily using for this. And I put this up here just to show the degree of conservation between yeast and higher eukaryotes. So all of the enzymatic activities are conserved between yeast and metazoans. Notable differences are PTP-MT1, which is phylogenetically unrelated to GEP4, but it catalyzes the same reaction. And the lipase for higher eukaryotes has not been identified, whereas it has been for yeast. Although we know based on the enzymatic function of tafazin that there needs to be some sort of lipase activity upstream of tafazin. And then, of course, in Barth syndrome, there's no tafazin activity, and it leads to the three biochemical consequences. So the accumulation of monolysocardiolipin, decreased total cardiolipin levels, and then also the cardiolipin that's there has abnormal acyl chain compositions. And one of these or any of these in combination are going to contribute to mitochondrial dysfunction and ultimately to at least part of the pathogenesis of Barth syndrome. And we really wanted to take a closer look at how the different acyl chain compositions of cardiolipin are able to affect mitochondrial function. So we asked the question, is the abnormal cardiolipin acyl chain composition really what's causing this dysfunction? Or to kind of rephrase this question, do you need a normal molecular composition of cardiolipin for normal mitochondrial function? And so there's a lot of sort of lines of thinking that suggest that this actually is important. And the first is quite simply that these pathways are conserved from yeast all the way up into humans. And this is just, again, showing side by side the two pathways. And in addition to being conserved, there are also additional proteins in higher eukaryotes that are also able to change the molecular species of cardiolipin, suggesting that this is important. And then the fact that throughout evolutionary time, there's been changes in the actual genes or the actual proteins doing this, but they have the same catalytic function, suggests that this is an important pathway to remain intact. The second is that there's altered acyl chain compositions in a number of diseases. The most relevant, I think, is Barth syndrome, but there's also been changes in acyl chain composition observed in various models of heart failure, in cardiomyopathies, in cancer, and so it seems like this is required for normal function for mitochondria. And then, based on Dr. Slami's work, there's this idea that different species have this different acyl chain composition. So within a single species or a single cell type, there is only a few different molecular species of cardiolipin, but between different species and between different tissues within the same species, they can have different molecular acyl chain compositions. And so if we take this last point and we combine it with the fact that not all mitochondria are the same, and this is just a figure from one of Vamsi Mutha's proteomics papers where they looked at the proteome of different tissues within a mouse, and the punchline from this is that different tissues have different proteomes, which suggests they have different energetic requirements and different mitochondrial requirements. If you combine that with the fact that different tissues have different cardiolipin acyl chain compositions, this has all kind of led to this hypothesis that the purpose of cardiolipin remodeling is to generate a specific molecular form, and that this molecular form of cardiolipin may be important for optimizing function or making sure that the mitochondria is specifically suited for the cell's needs. Now, the important question that so far hasn't been properly addressed, we don't think, is that do these different forms of cardiolipin actually have different functional capacities? And this really, there haven't been good models to test this in otherwise isogenic cells. But with the identification of CLD as the lipase, we think that there may actually, this may be a good model to do this. So if we can block CLD function, we can stop remodeling, and this may give us a good model to actually test this. And so we went about verifying this, was that me? Okay. So we wanted to verify that this delta CLD yeast mutant is actually going to be a good model to test this. And so it really isn't projecting well here, but these are radiolabeled phospholipids separated by TLC. And in the wild type situation, there's a normal amount of cardiolipin. In the cardiolipin synthase mutant, there's no cardiolipin at all. In the CLD mutant, there's a normal amount of cardiolipin similar to wild type, which suggests that if you mutate or if you disrupt cardiolipin remodeling, at least at its initiation point, is not affecting cardiolipin biosynthesis at all. So you can still make cardiolipin to the same level as you can in wild type. In the tafazin mutant, as expected, there's a decrease in cardiolipin, the accumulation of monolysis cardiolipin. And in the double mutant CLD tafazin yeast strain, there's no monolysis cardiolipin, and the cardiolipin is back to wild type like levels, which verifies that this is kind of a linear pathway. It also indicates that CLD is the only lipase that works upstream of tafazin. So we wanted to look a little bit more closely at the molecular composition of cardiolipin, and we did this in collaboration with Jinlin Han at the Stanford Burnham Research Institute. And he looked at these different strains by mass spec and categorized them by their molecular species. And so we have here at the bottom just all the different molecular species of cardiolipin, and then their relative abundance in wild types in black, delta CLD in yellow, delta tafazin in blue, and then the double mutant in green. I hope you can at least appreciate from this, although it's very complicated, that the different strains have different, a different pattern of molecular compositions. And so to look at this in a way that's a little bit easier to digest, we categorize the molecular species by the amount of unsaturated acyl chains. And so in the wild type cells, 92% of all cardiolipins have 4 unsaturated acyl chains, and there's only 8% that have 1 saturated acyl chain and 3 unsaturated acyl chains. This is in stark contrast to the delta CLD mutant, where only about 20% of the cardiolipin has 4 unsaturated acyl chains. The rest have at least 1 and sometimes 2 saturated acyl chains. In tafazin, the overall abundance of cardiolipin is a little bit lower. There's still a similar amount of cardiolipin that looks completely remodeled that has 4 unsaturated acyl chains, but the overall levels are rescued in the double mutant. So in delta CLD, we have altered acyl chain compositions, but we have a normal amount of cardiolipin. And so we can really use this as a model to specifically test the relative contributions of these different molecular forms to mitochondrial function. And so cardiolipin is involved in a whole host of different mitochondrial functions. In the data that I'm going to show here, we're just going to focus on oxidative phosphorylation. And this is in part because there are a lot of really sensitive tests that we can use to measure this, and also because cardiolipin's role in oxidative phosphorylation is very well characterized. We also, in data that I'm not going to show today, looked at the mitochondrial morphology by EM in collaboration with Mike McCaffrey, but I will not be showing that today. So the first thing we did is look at the formation of supercomplexes. And in yeast, this is characterized, the supercomplexes are composed of 2 copies of complex 3 and 1 or 2 copies of complex 4. And this is analyzed by Blue Native Page, so we're separating intact protein complexes by size. And so in wild type, this is a blot for a subunit of complex 3. This higher band represents 2 copies of complex 3 and 2 of complex 4. The lower band in the wild type represents 2 copies of complex 3 and 1 of complex 4. In the absence of cardiolipin and the cardiolipin synthase mutant, there's an altered stoichiometry between the 2 supercomplexes. So there's an increase in the abundance of the smaller one, and then free complex 3 also appears. And the same pattern is seen with blots for complex 4. So there are 2 different, in the wild type, there are 2 different supercomplexes. The stoichiometry is altered in the delta CRD strain, and free complex 4 is seen at the bottom. So without cardiolipin, these supercomplexes really break apart. In complex 5 in the yeast can exist as a monomer or a dimer, and this really isn't affected by the presence or absence of cardiolipin. And then we also looked at the assembly of AAC. AAC is the ADP-ATP carrier on the mitochondrial inner membrane. It associates with respiratory supercomplexes, as seen up here, but it also associates with a number of different proteins. And in the absence of cardiolipin, these complexes are really disassembled. So filling out the rest of the panel, and we'll just focus on the complex 4 blot. Looking at the delta CLD strain, it looks like wild type. So there's a high abundance of this larger supercomplex, not too much of the smaller supercomplex, and there isn't any complex 4 that's freely at the bottom of the gel. In the delta tafazin strain, there's in our particular strain, it looks similar to wild type, although there is a little bit of free complex 4 that appears here. And importantly, the CLD tafazin strain looks like CLD and looks like wild type. The defect in tafazin is a lot easier to see in the AAC blot, so the association of AAC in these larger molecular weight ranges, which represent its association with supercomplexes, is reduced a little bit in the absence of tafazin, but it's rescued in the CLD tafazin double mutant. All of that is assembly, and so we also looked at the just protein expression of the various oxidative phosphorylation subunits, and this was work that was completed by Yawen Liu. And the punchline here, this is a lot of different blots. There's no real difference in the actual protein expression for any of these different proteins. So we also, all that data suggests that the acyl chain composition isn't important for protein expression or for the assembly of supercomplexes. We also wanted to measure oxidative phosphorylation function. And so the way we did this was, or one of the ways we had done this was measuring oxygen consumption. Remember that complex 4 is the final step in the electron transport chain. It reduces oxygen to water. So we can take intact mitochondria, put them in a chamber with an oxygen electrode. If we add a phosphate and a substrate, then there's some sort of basal level. This is called state 4 respiration. If we add ADP, this stimulates the ATP synthase, utilizes the membrane, or utilizes the electrochemical gradient, and allows for faster proton pumping, increase in the electron transport chain activity, and so this leads to a bigger or a faster rate of oxygen consumption. And then at some point, if you had a limiting amount of ADP, this is going to run out and we'll return to state 4. So there are two really important measurements you can get out of this. The ratio of state 3 respiration to state 4 is called the respiratory control ratio, and this gives you an indication of how well coupled oxidative phosphorylation is. And you can also measure the amount of oxygen that is utilized during this step relative to the amount of ADP, which was added, and this is the P to O ratio, and it gives us a really good idea of how well coupled oxidative phosphorylation is. So when we did this analysis in the different strains, in the absence of cardiolipin, the RCR value is really decreased compared to wild type, but in the CLD mutant, it seems to be the same as wild type. In the tafazin mutant, there's sort of an intermediate defect, so it's not as bad as the complete absence of cardiolipin, but it's still not to the level of wild type. And the double mutant looks like CLD and also looks like wild type here. That was the RCR value, so that's oxidative phosphorylation coupling. Looking at the P to O ratio, which is oxidative phosphorylation efficiency, this is essentially the same story. So a defect in the cardiolipin synthase mutant, less of a defect in the tafazin mutant, no defect at all in the absence of the CLD. Looking at this in mitochondria that are uncoupled, so we disrupted the membrane potential, and this allows the electron transport chain proteins to function at a maximal rate. There's a little bit of a defect in the absence of cardiolipin, and we had gone on to do further studies to kind of separate complex 3 and complex 4 activity. This defect seems to be in complex 3, which is entirely consistent with the role of cardiolipin in its catalytic cycle. But again, there's no defect in the absence of CLD. We wanted to do this using a second method, and so this was in collaboration with Nate Alder at the University of Connecticut, and he measured the membrane potential of these different yeast strains. And so the way this works is that you have a dye that is fluorescent, and when mitochondria hyperpolarize, this dye moves into mitochondria, and it self-quenches. So what we're looking at is a decrease in fluorescence intensity, which corresponds to an increase in the membrane potential. So we can take mitochondria, add a substrate, it starts the electron transport chain, and you get hyperpolarization of the membrane. If we add ADP, we can utilize the membrane potential, causing a transient depolarization. This is state 3 respiration. At some point, the ADP is used up, the membrane repolarizes, and this is state 4 again. Filling this in with all of the rest of the strains, it's a little bit busy here, but wild type and CLD both follow this same trace down here, whereas in the absence of cardiolipin synthase, there's a slower return to state 4, and in the tafazin strain, there's a slower return to state 4. And we were able to quantify that here, which shows just that. Wild type and delta CLD are essentially indistinguishable, and there's defects in cardiolipin synthase mutants and the tafazin mutant. And this is just growth on respiratory media. This is not nearly as sensitive as the other studies that we had done, but it's all consistent. So looking just on the ethanol glycerol plate at elevated temperature, wild type is able to grow, delta CRD is not, delta CLD is able to grow, delta tafazin cannot grow, and the double mutant phenocopies delta CLD and phenocopies wild type. So all of these data together suggest that despite, we can measure the assembly of supercomplexes, we can measure individual complex or subunit expression, we can measure different aspects of oxidative phosphorylation function, but whether the strains have a wild type or a normal molecular composition of cardiolipin, or if they have an altered molecular form of cardiolipin in the delta CLD mutant, these are indistinguishable from each other. So this suggests that it's actually not, at least in these strains, and at least in yeast, the acyl chain composition doesn't make that much of a difference in terms of oxidative phosphorylation. And so we were a little bit puzzled by this, but it's kind of made us take a step back and sort of reanalyze how we're thinking about this. And the one thing that I think is particularly relevant here is the implications of this for Barth syndrome, right? So in Barth syndrome, you have the 3 biochemical consequences: increased monolysis of cardiolipin, decreased cardiolipin, and the altered acyl chain composition and mitochondrial dysfunction. If we delete CLD or inhibit CLD, even in the presence of a tafazin mutant, there's no monolysis of cardiolipin accumulation, there's normal levels of cardiolipin, despite the fact that they still have an abnormal acyl chain composition. And oxidative phosphorylation, at least, and in data that I didn't show, mitochondrial morphology is normal in these strains. So potentially whatever in humans is participating in this lipase activity could be some sort of therapeutic target, provided that it's actually identified. But in sort of a broader sense, and more of a basic biological sense, the question is really, what's the physiological role of cardiolipin remodeling? And this, I think, is consistent with what Dr. Greenberg had as her second hypothesis, which is one of the things that we had come up with. So the electron transport chain is the major source of cellular reactive oxygen species in a cell. And so this ROS can cause a lot of damage and cause a lot of dysfunction. And so cardiolipin is specifically associated with all of the members of the electron transport chain. And so the idea is that maybe instead of establishing a specific molecular form of cardiolipin, instead this pathway exists to remove and replace acyl chains and potentially damaged acyl chains. And so this is something that I think makes sense with some of the data that's out there. So in yeast lacking tafazin, as well as Barth syndrome patient cells, and in the mouse model, these all have increased oxidative damage. So we think it might fit in kind of that realm. And then also, it's important to remember that all the studies that we had done measuring oxidative phosphorylation were in optimal conditions. So it could be that if we kind of stress them out a little bit with something that would induce reactive oxygen species or something of that nature, that potentially we could distinguish between wild type and delta CLD. And so as we were kind of thinking about this, there was something that was a little bit strange to us that we've affectionately called the paradox of the repair model. And that's simply that in general, unremodeled cardiolipin has more saturated acyl chains. Remodeled cardiolipin has unsaturated acyl chains. And so if this is really a repair mechanism, why would the process of repair put on unsaturated acyl chains that are more susceptible to oxidative damage? And so in thinking about this, we've come up with something that's absolutely hand-waving at this point, but we think it's an interesting idea, at least, in that cardiolipin, because it's closely associated with the electron transport chain and because it has a dedicated repair mechanism, potentially, that maybe this acts as a buffer or a shield that can actually absorb the reactive oxygen species and potentially protect oxidation of other components in the mitochondria and other components in the cell. So by having these unsaturated acyl chains next to the ROS producers, it prevents the ROS from getting to mitochondrial DNA, prevents it from getting to other mitochondrial proteins. And this is all consistent with some work we had done with the regulation of CLD regarding the membrane potential. So they've shown in vitro that if cardiolipin has oxidized acyl chains, there's a decrease in the function of complex 3 and complex 4. So potentially, when that happens, there's some sort of signal, like a decrease in the membrane potential, which will increase the rate of cardiolipin remodeling, removing more acyl chains, putting on more new acyl chains, and kind of returning this to homeostasis. And so with that, I'm going to thank I did all the work in Steve Claypool's lab. Yawen Liu did the protein expression analysis. She also helped with the blue native analysis. She also has a poster that she'll be presenting later today. And then our collaborators, Jillian Han, Nate Alder, and Mike McCaffrey. Thank you.
Thank you, Matt. It was very inspiring. I've enjoyed your talk. I'm going to ask you a tough question, but before I ask it, I'm going to invite Miriam and Mindong to comment as well, but you get first crack at it.
Okay.
So I think both your group and Miriam's group have shown very elegantly that remodeling of cardiolipin doesn't matter in yeast, but skeletal muscle, striated muscle, is different from yeast, and I wonder, is that true in striated muscle? Do you think this is generalizable to that? And then, has anyone done the experiment, or is there a CLD1 ortholog in Drosophila, and would it rescue the fly phenotype?
Yeah, I mean, it's an interesting question. We don't know the lipase involved. So in Drosophila, Dr. Salami has been able to knock out one of the phospholipase A2s and have a partial rescue of dephasing flies, at least in the cardiolipin content. But it's not a full rescue. It's certainly not what we see in yeast. And so there's potentially other lipases that are participating in this process that we don't know of.
In Drosophila, there is no homolog of CLD1, and so that's the answer. And also about the tissue distribution of the molecular species. So in the heart, remember the previous talk we had by Gilliam, and he mentioned in the ACSL knockout, there you have a completely almost a depletion of tetraalyl cardiolipin, but there's no heart failure happened there. So it seems sort of consistent with the yeast results here.
Well, I'd like to, oh, did you want to say something, Mary? It's a very interesting hypothesis that you present about the reactive oxygen species, and perhaps it's valid, but I'd like to think about it a little bit differently in that what may be happening is that there's an evolutionary process where tetralineal cardiolipin is a preferred environment for the electron transport chain, and when a tissue is under stress, such as the heart or skeletal muscle in an active oxidative environment, then you're going to see the defect. So when you do what you do in yeast, and you now have the non-tetralineal composition, you don't see a manifestation of that, but only under a specific stressed environment, such as in the cardiac tissue and working skeletal muscle tissue, in much the same way that brain, we don't see anything.
Yeah, I mean, that's absolutely possible. We can't do those experiments in heart or brain. We're working with the best model that we have available right now, but that's absolutely a valid point. It certainly could be the case. But based on what we had seen, we wanted to start thinking about alternative explanations for what this pathway is actually doing, just because our data so far in yeast doesn't suggest that the acyl chain composition is important.
For basal oxidation.
Sure, absolutely, absolutely.
I just would like to agree with both of your points, that when we look in yeast, we look in the absolute optimal growth condition, or we can add stresses as we see fit. But the real problem is we don't really know. I mean, growth in the laboratory at 30 degrees in all the right nutrients is not exactly what the yeast cells experience out on the vine, in real life. And similarly, what's really going on in mammalian cells. So I think that at least one big advantage that we have in the yeast system is that we can do a genetic analysis which is really unbiased, and we can say under what condition is there a detriment to not having this phospholipase. That's what we're doing with the SGA analysis, and I think that will get to the question.
Are you answering or commenting on that? So I wanted to maybe make a comment in the same direction, that somehow in mammalian cells, tetralineal cardiolipin with two double bonds, with this bis allelic hydrogen that can be abstracted, that has been what has been chosen by Mother Nature as this optimized cardiolipin. So now your lipidomics analysis that Shanling Han has performed for you clearly indicates that you don't have linoleic acid containing species, or
No, not in yeast. Everything predominantly has one unsaturation.
but then
right?
I wanted to indicate to you and to everybody that oleic acid containing cardiolipins are not readily oxidized at all, and for this reason this Ross hypothesis is kind of hanging around there without being directly applicable, because it's very difficult to oxidize oleic acid. There are only some forms of P450 that can perform this hydroxylation.
Yes, I think this is actually a very important point. This often gets forgotten by biologists, that monounsaturated fatty acids are not really oxidizable unless you put them into gasoline and throw a match in it. So there is, yes. The other thing I wanted to add is something that has sort of not been mentioned here, which is very important. Consider the possibility that it is not the composition of cardiolipin that is important, but the actual process of remodeling. It may be involved in assembly, and that goes very much, I'm basically is in support of what your data have shown.